Hello and welcome to the Biomedical Informatics and Data Science Grand Rounds here at Johns Hopkins University in the School of Medicine. My name is Paul Naji, and it's really a pleasure to introduce Dr. Tiffany Callahan, who will be our Grand Round speaker today. Tiffany, had received her PhD in computational biology from the University of Colorado, with her thesis topic on leveraging graph representations, learning and neural symbolic reasoning of large-scale biological knowledge graphs in order to develop realistic estimates of human disease mechanisms. And she has also served, she is currently a postdoctoral research scientist at IBM, and she's also been a postdoctoral fellow at Columbia University. One of the things that really excites me about the work that she's doing is that she's been working with the OMA Common Data Model, which by definition is a very conventional data model. It's using relational SQL, and she's been able to connect this very relational database to be able to work with biomedical ontologies. And this allows us to have the real flexibility to work in a multimodal sense, even within the OMOP data set. So Tiffany, thank you so much for coming today and presenting to us your work.
Thank you for the great introduction. I'm really excited to be speaking with you today about some resources and tools that I've been developing sort of towards the ends of my dissertation. So I have no conflicts of interest to disclose. After attending this presentation, the participant should have the ability to explain the value of common data models for conducting biomedical research, describe what a biomedical ontology is and how it can be applied to improve patient care, identify barriers and solutions to aligning clinical codes like diagnoses and lab tests to open source biomedical data like ontologies and knowledge graphs, and to understand how knowledge graphs can aid in clinical phenotyping, specifically within the case of identifying undiagnosed rare disease patients. I just want to mention that the work presented in this talk was primarily conducted during my PhD at the University of Colorado and as part of my postdoctoral fellowship at Columbia University. The work was funded by 2 training grants from the NIH, specifically the National Library of Medicine. And it's important to note, since I am at IBM now, that any opinions, findings, and conclusions or recommendations expressed in this talk and any of the associated materials are mine and do not necessarily reflect those of my current employer. Okay, on to the fun stuff now. So this is a very brief, high-level outline of the talk that I'll go over today. And we'll cover 3 topics. So we'll start by reviewing relevant background, and then we'll jump right into 2 studies that sort of demonstrate and solve problems that we mentioned in the background. So to ensure that we're all on the same page, let's get right into the background part. So in case you're not familiar, clinical phenotypes are specifications of an observable state of an organism, in contrast to a genotype, which is derived from an organism's genetic makeup. Computational phenotypes can then be thought of as machine-readable versions of these specifications, and most often they're applied to EHR data. The figure on this slide provides a really simple example of a computational phenotype designed to identify patients with acute kidney injury. And deep phenotypes we can think of as algorithmic solutions to improve computational phenotypes by incorporating additional data. And we do this because we want to create more accurate and precise descriptions of a patient's state. But there are several challenges with developing and using existing computational phenotypes. So traditional development techniques tend to result in very accurate patient cohorts, but this comes at the cost of requiring a significant amount of resources to develop. And due to the nature of EHRs and the manual development process, existing phenotypes often have limited interoperability and portability. Traditional approaches are based off of our current knowledge, which also often presents this limitation to us only being able to capture well-known or well-characterized phenotypes or diseases. So given these challenges, there are several open questions that I thought if we could address could significantly improve the utility of computational phenotypes. So given all of the data modalities available to build computational phenotypes, which should be prioritized? And is this decision dependent upon the phenotype of interest? And the second question is it possible to develop algorithms that can accurately identify patient cohorts and provide some sort of causal explanation as to the resulting phenotype or disease state that we observe? So to address the first question, we conducted ablation studies designed to examine the impact of patient representations that were built using different combinations of data types and sampling windows. To address the second question, we compared patient representations that we built using only clinical data to those that we built using knowledge of the molecular mechanisms that underlie human disease. So let's jump into that first question. A little background first. So representation learning techniques are a way to meaningfully transform a wide range of complex, heterogeneous data into representations that are well suited for a wide variety of downstream learning tasks. And I always like to show a simple example, which I'm showing in the figure at the bottom of this slide, of how these methods work. And so data for a patient is aligned by first their visit date, and then we treat the events that occur at a given visit as sentences so that we can derive an embedding for a specific diagnosis. In this case, we want to know what it means for this patient to have asthma. So the objective of our first study was to perform an extensive data-driven characterization of patient representation learning methods for the purpose of automating computational phenotype development. Our primary question was, what combinations of data type and sampling window create the best patient representations, and does performance differ by disease? To answer this question, we conducted ablation studies designed to examine the impact of patient representations built using different data types and different sampling windows. Because we had access to validated phenotypes that we could further verify with newborn screens, we performed our experiments on a sample of rare disease patients. And the specific rare diseases we selected were chosen because they have aspects which make them distinct, but also they share some symptoms which should make phenotyping a bit more difficult. In addition to the rare disease patients, we also selected a random sample of medically complex controls, so 10,000 of them. Patients were represented using a very simple yet performant method, bag of words with TF, IDF. We built several representations varying the sampling window based off of an index date, where the index date in this case was the earliest date of diagnosis relevant to the primary disease, or the earliest date of any kind of diagnosis for control patients. We included all concepts before and after the index date, as well as a combined view of both before and after concepts. And we also varied the data type, where representations were built using either only conditions, only medications, only measurements, or all of those data types. Representations were evaluated using a series of experiments where the impact of altering different data elements was assessed by how well each resulting representation was able to accurately identify similar patients of a specific disease type. So first we used a Uden index, which is a receiver operating curve summary statistic that takes a continuous variable, in this case, a similarity score between two patients, as an input, and outputs a diagnostic threshold, and then we perform leave one out classification. So we clustered the representations using k-means with a predefined k of 4, because that's how many different disease groups we had. And in this case, only assessing rare disease patients, or 5 when we included control patients. For all experiments, we evaluated performance using, again, only rare disease, and then rare disease plus control patients. So we wanted to have, control patients have a lot of variance and a lot of noise. So we wanted to see if we can do a good job with just rare disease patients. What happens when we add in the control patient? This plot shows, captures the performance of patient representation for each rare disease by data type and sampling window, where the data types are grouped within each disease, and the 3 plots represent the different sampling windows, so before, after, and all concepts. So in general, what do we see from this? Regardless of the metric we use, using concepts after the index date or all available concepts tends to work better than using only concepts that occur before the index date. But let's take a closer look when we actually see what happens when we dive into specific diseases and the data types within them. So this slide displays the performance of one of our rare disease groups, in this case, PKU patients. And columns are data types and rows are sampling windows. So as shown in this slide and highlighted by a red arrow, the best performance for this group of patients was achieved when using concepts from all domains, so conditions, medications, labs, all together, but only those things that occurred before the index date. So in general, we can see that some domains perform better than others, regardless of the sampling window, In this case, using all domains is best, and using medications, no matter which window you use, tends to perform the worst. So this is an interesting pattern to keep in mind as I show you the other rare diseases. So now we're looking at cystic fibrosis patients, as shown in this slide, and again highlighted by the red arrow, the red arrows, two of them. The best performance was achieved when using all concepts from all domains. And in general, we can see that regardless of the clinical domain, concepts before the index state tend to do very badly compared to looking at things that happen after an index state or when we're looking at everything together. But in general, remembering the pattern you saw from PKU patients, we do better with medications for this group than we did for PKU. Now we're looking at sickle cell disease. The best performance, also shown by red arrows, was achieved when using condition concepts or all concepts from all domains. And in general, we can see compared to the prior two diseases, performance, no matter which window or which data type, is significantly worse than the other two disease types. So what is that telling us? It's really hard to classify patients based off of representations of their codes when we're trying to really identify sickle cell patients. And the final group, congenital hypothyroidism, the best performance was achieved when using all domain concepts or all concepts after the index date. And in general, we can see regardless of the sampling window, in this case, all domains perform similarly well, but we also, just like sickle cell disease, compared to cystic fibrosis and PKU, they're difficult patients to identify, and this could be because they're difficult to continue to diagnose, or they have things that change frequently over time that make them look like the other disease groups. But just using clinical data alone, regardless of the window or the data type, isn't necessarily super effective. So these plots display the best performing representation when clustering only rare disease patients. On the left is a t-SNE plot, and on the right is the same plot, but this time colored by clustering assignment, when K was set to 4. So again, looking only at rare disease groups. So for each of these clusters, the top 3 most frequent concepts and the top 3 most frequent unique concepts for each cluster were reviewed by a clinical expert who confirmed that we were able to not only capture the most important diagnoses, but that these were also the most important features that distinctively described these patient types. So overall, we did fairly well at separating them. Many of the important features used in traditional phenotyping for these diseases were not identified among the most important, so we actually kept those out. And this confirmed our suspicion that not all of the concepts included in traditional phenotype definitions may actually be required to identify relevant patients or to distinguish them from each other. So wrapping up this first study, what did we learn and what did we demonstrate? So we learned that there is no single best combination of sampling window or data type that performs equally well for all diseases. The best patient representation then is likely to depend on the use case and the disease that you're looking at. And only a portion of all available data and data types might be needed, so these traditional definitions, which have very specific sets of diagnosis codes, might not actually be the best way to find your patients of interest. So what's our major takeaway? Accurate and performant patient representations require the right combinations of data elements, and this can be enhanced and improved by insight that can be derived from these data-driven characterizations. So you might not know ahead of time what works best for each patient group, so it's worth doing some of these shallow characterization tasks before a bigger learning task to really understand, how do I find these patients and what do I really need to do that well? Okay, moving on to the second study. So knowledge graphs are a tool that are frequently used to represent complex relationships, and they usually are built with a wide variety of heterogeneous data. Knowledge graphs can also capture causality, most often in the form of directed networks where an edge between 2 nodes usually indicates some sort of causal relationship. While there are many applications of knowledge graphs in the biomedical domain, there are 2 that are particularly important for this second study. First, observational data sets like EHRs are really only able to capture associations between data that are present in your database. Knowledge graphs can be used as a proxy for phenomena that may otherwise be missing or difficult to explain when you're limited to only one type of data. or one data source. In addition to capturing missing information, knowledge graphs can also be used to provide causal explanations of known phenomena and even help us generate novel hypotheses. The goal of the second study was to determine the clinical utility of patient representations that we build or that are built from a giant knowledge graph. To do this, we compared patient representations that were derived using only clinical data to those that were built using a knowledge graph that we built with the intention of representing molecular mechanisms underlying human disease. So before diving into these results, I want to introduce you to two resources that we had to build in order to conduct these studies. The first one we call Phenolater, which I really like the name of because it reminds me of the candy Now and Later, but it actually has a meaningful name. It stands for Phenotype Knowledge Translator. And what it is a semantic ecosystem for automating the fair construction of ontologically grounded knowledge graphs with fully customizable knowledge representation. This figure provides a very high-level overview, sort of the roadmap of our ecosystem, and it highlights three components, which are shown in the three different gray boxes. So why are these important? What are these components and why do they matter? So the first one is knowledge construction resources. So it includes resources and tools to help download and process data, custom algorithms to help construct large-scale knowledge graphs. It also contains knowledge graph benchmarks. So when we build knowledge graphs, we like to keep them. So we consider these pre-built knowledge graphs that have all of the same data, but have different modeling decisions for how we put nodes together. And these are important because they can help us to systematically assess the effects of using different types of knowledge representations on specific downstream tasks, like workflows and different algorithms. And then the final piece of this ecosystem are tools, specifically tools to use knowledge graphs, so things like cloud-based data storage, APIs to help access our data and facilitate accessing other people's data, and triple stores to look at the graphs and query them. This figure represents the knowledge representation that we used for this study. So again, our goal was to take the knowledge we have of biology all the way from the central dogma up to human observable disease to construct this graph, which really, we hope, represents mechanisms of human disease. So to be a proxy for if you had actual molecular data on patients. The purple box represents experimental data. The blue box represents molecular mechanisms that we created by integrating different open biological and biomedical foundry ontologies, which are also shown in gold and green. Edges between ontologies are created by integrating other data sources that are not originally part of them. Dashed lines represent relationships that are inferred from the relation ontology, and dotted lines represent relationships that already existed between the ontologies that we merged together. So the final knowledge graph used in study 2 was constructed from 12 different OBO ontologies, 22 linked open data sets, 2 experimental data sets, and it ended up containing over 700,000 nodes and over 25 million edges, so not a small graph. The second resource that we had to build to do these studies, we've named OMOP to OBO. This is a system that generates mappings between standard OMOP vocabulary concepts and OBO ontology concepts. So the system obtains concepts and metadata from both OMOP and OBO, and it tries to obtain a mapping by first directly aligning concept codes and strings, and then leverages concept embeddings to help match concepts using things like string labels and synonyms. If a match is unable to be obtained from any of the prior approaches, it also includes a component that can take manually annotated mappings. And throughout this whole mapping process, when you're running this algorithm, the system tracks important metadata and collects evidence that it uses to categorize the mappings. So what does that mean? So when you get a mapping out of it, you should know exactly what it was from either source that was used to create that mapping. Was it a OMOP synonym that connected to a actual concept in OBO, or was it that they were aligned based on their definitions that we generated from embeddings? And so we leverage this system within study 2 to help us map a large subset of condition occurrence, drug exposure ingredients, and measurement results. And before diving into those results, I just want to give you a feel for what these kinds of mappings look like. So starting with condition occurrence, We mapped them to two OBO ontologies, the human phenotype ontology and the Mondo disease ontology. And the examples on the screen show one simple high confidence mapping from each ontology, in this case for heartburn and for Weber-Gubler syndrome. Drug exposure ingredient concepts were mapped to four OBO ontologies. Chemicals were mapped to KEBI, organisms were mapped to the NCBI taxon, proteins were mapped to the protein ontology, and vaccines were mapped to the vaccine ontology. OMOP concepts were mapped only to those ontologies that made sense, so you didn't have to map to all four of them if that didn't make sense for the OMOP concept. And that's nicely generated, I think, by the three examples we show on this slide. So for example, idosglucan is mapped to three ontology concepts. It's both a human antineoplastic agent that involves the human protein interleukin-2. Measurement concepts are mapped by interpreting their results against a reference range, where the result is aligned to the human phenotype ontology and the constituent parts of the lab. So like for the drug example, drug exposure ingredients, they're mapped to several other ontologies. So two examples are shown on this slide. This first demonstrates what a positive and negative result for a urine test for amphetamines would look like. Here we use logic on the human phenotype ontology concept to indicate a positive or negative result. We map urine to the Uber anatomy concept for urine, and we map amphetamines to the KEBI ontology concept. So for study 2, we used OMOP to OBO to build mappings for over 92,000 conditions, 8,000 drug ingredients, and 10,000 measurement results, 2 ontology concepts, which represented over 9,000 diseases, over 6,000 phenotypes, 83 different anatomical entities, 2,700 organisms, 4,000 chemicals, 132 vaccines, and 272 proteins. So this is a nice slide to show you what we started with and what we mapped them to in terms of ontologies. And again, these ontologies are sort of the grounding stakes within our knowledge graph, which we then make better or elaborate by using other external resources. So in order to get from OMOP to the knowledge graph, we need OMOP to OBO to help us get there. And we build the knowledge graph using Phenolater. Okay, so how do we know that our mappings are useful or good? Just to give you some confidence before we dive back into study 2, we evaluated them in 3 different ways. So first, we looked at the accuracy of the mappings, where we consulted with 10 different domain experts and asked them to manually review 20% of the most challenging manual mappings. So the automated mappings, they all have confidence scores with them, and they're all derived from the actual resources that we use. We can feel, because we trust the OMOP community, we trust the OBO community, we can feel that we trust the alignments of resources that they provide. What we're most concerned is, what about the ones that we had to manually do? So for these, we found correct mappings for 74% of conditions, 71% of drug ingredients, and 93% of measurements. So fairly good overall. And we used what we learned from this to sort of back update the mapping process and help us do better in the future. The second way we evaluated them was for generalizability, meaning we wanted to know how useful the mappings would be to researchers at other institutions. So if I gave you these mappings today, how likely would it be that they would cover concepts that are relevant to your analysis? So to evaluate them, we compared the coverage of our mapping set to 24 different hospitals, those that were part of the Odyssey concept prevalence study specifically. And we found that 99.5% of conditions, 99.9% of drug ingredients, and 60% of measurements used at least once in clinical practice across these institutions were covered by our mappings. Finally, we evaluated the mappings on their clinical utility by comparing them to a validated manual set of mappings when used to identify patients with rare genetic diseases from the All of Us research program. And we found that the OMOP to OBO mappings were able to identify 99.3% of the patients that the other set of mappings identified, but they were able to do this using fewer codes and in 1/3 of the query time. So fewer codes and faster. So we felt, hopefully this gives you some confidence that these mappings are accurate, they're useful, so they cover concepts that are generally used in other institutions besides the ones we built them from, and that they are clinically meaningful in terms of being able to find patients that you intend to find when compared to a gold standard set of mappings. So study 2. So we use the same set of pediatric rare disease patients that we used in study 1. And similar to study 1, patient representations were built using different combinations of clinical data. And in addition to the bag of words model, which we used as our baseline, we also derived representations using Gach-de-Beck. So we built representations in 2 ways from the phenolater knowledge graph, again, designed to represent molecular mechanisms underlying human disease. To build the clinical patient representations, we aggregated concept embeddings for each patient's set of conditions, drugs, and labs. To build the molecular mechanism patient representations, we aggregated node embeddings derived from the knowledge graph for each patient's unique set of conditions, drugs, and labs, which were mapped to node embeddings using OMAP to OBO. To evaluate the representations, we performed one versus the rest multi-class classification. We also conducted a case study, which included an independent sample of pediatric patients that had been diagnosed with sickle cell disease, who had gene expression data, a measure of sickle cell severity, and a sickle cell disease genotype. For these GEO patients, so that's where we obtained this data set from, we aggregated the node embeddings for each of their genes from our knowledge graph, which were first adjusted by each patient's unique molecular signature, which we obtained directly from their gene expression data. To evaluate the patient representations and identify genotypes for each of the sickle cell patients, we used different patient similarity-based analyses. So this slide provides a high-level overview of the process that was used to create the patient representations. For representations created using only clinical data, we aligned patient concepts by visit, treated them as sentences, and then embedded them. For the molecular mechanism representations, we mapped clinical concepts to nodes in the phenolater knowledge graph, and then used deep walk, a node embedding algorithm, to extract paths of relevant mechanisms for each node. So the example that you're seeing here on the screen is for asthma. Well, what does asthma mean when we're pulling information from our knowledge graph? Well, asthma might have a path that looks like the one shown on the screen. So it may have things like albuterol and prednisone, which are substances from the knowledge graph that we know treat asthma. It may have cough, which is a symptom that we know belongs to the asthma phenotype. And that symptom is caused by the mucus secretion pathway. Cough also has a phenotype, a gene associated with it, ADBR2, which is known to cause or contribute to asthma. Same with HLA-G. HLA-G also has a phenotype, atopic dermatitis, which we also observe with asthma. So you can see, compared to the clinical representations, which are shown above the dashed line, the knowledge graph mechanism-based way of representing patients is really more about the context of what we know about disease at different levels. So what do we know at the genetic level? What do we know at the pathway level? What kinds of symptoms or phenotypes would we expect to be associated with this diagnosis? So you're getting more context, which could either be very useful to help you identify patients where information is missing from the clinical record, or it could add extra noise that might not be relevant to the actual patient that you're looking at. So the task here was really to understand, how do these representations differ? What knowledge do we get from one that we don't get from the other? And is it important enough that we'd want to pick one way of representing patients and not do the other one? So we ran many models, like hundreds of models, looking at different hyperparameters, looking at different representations. And what I'm highlighting for you here are the t-SNE plots for the best performing models when using only clinical data. Those are shown in the 2 columns on the left, versus the knowledge graph molecular mechanisms, the 2 columns on the right. In each of the 4 sets of plots, moving from left to right, then top to bottom, we feature representations built using only conditions, only medications, only measurements, and then all of those things combined. The 4 top plots were built using only rare disease patients. That's why you don't see any yellow dots in them. And the bottom plots are all the same information as the top, but in this case, we include control patients. I really like these plots because I think they nicely visualize how the representations differ by data domain and patient type. So for example, while both types of representations are able to group and sort of separate the rare disease patients by their disease type, the groupings from the knowledge graph mechanisms tend to be a bit tighter and almost more distinct or separated than what we can do when we're looking at only clinical data. And this interpretation was supported by the actual modeling performance metrics as well. But let's dive in a bit deeper. So this side is just focusing in on the single best parameterization of the models for only clinical data, shown in the 2 columns on the left, or molecular mechanisms from the knowledge graph, shown on the right. And again, we're showing you the same t-SNE plot on top. On bottom, we've just colored that plot by cluster assignment. And so as you can see from this, the molecular mechanism-based representations produced more clusters, which were deemed to be more clinically relevant when we actually reviewed the top concepts with our domain experts. So for example, clusters 1 and 5 on the right side were able to separate out 2 clean groups of cystic fibrosis patients. When we dive into, well, what are these 2 groups? They end up being 2 of the most common types of cystic fibrosis based on their predominant symptom types. And in anticipation of the sickle cell disease specific case study, which I hinted at the beginning of study 2, I'm going to go through some of the most important features that we observed for each cluster just for the sickle cell disease patients. So the table on the left represents the count of concepts, total and unique, and patients identified for each cluster. In this case, I'm only going to present the results for sickle cell disease patients, for which we identified 3 subphenotypes from that prior figure. And this is just for the best performing clinical model first, so not to the knowledge graph yet. So what do we find? So we found Lyrica, which is a gabapentinoid, which is commonly prescribed to sickle cell disease patients to treat neuropathic pain. Mucopolysaccharidosis was also found to be important, and it's known to cause the accumulation of microparticles due to the inability to break down long-chain sugar molecules. HBSC, so that's one genotype or phenotype because it's Mendelian for sickle cell disease, one of the observed phenotypes. Patients with it tend to have lower levels of it in their bloodstream compared to patients with the more severe HBSS phenotype. And so what you're seeing here is this additional context. So we had to get this from the literature, but these kinds of explanations to justify what were the most important concepts we saw in this first cluster, that's something we're hoping we can get from the knowledge graph. But before we get to that, let me show you the next 2 clusters. So the second sickle cell disease cluster, again, using only clinical data, it showed sildenafil citrate or Viagra. We know from the literature that this drug has been shown to prevent priapism in sickle cell disease by increasing the degradation of serum nitric oxide. Urocidal is also commonly prescribed to sickle cell disease patients suffering from hepatobiliary complications, so things like peptic crisis, cirrhosis, or cholecystitis. And the final clinical sickle cell disease subphenotype we observed, we saw erythromycin. So this is used to provide pneumococcal infection prophylaxis in sickle cell patients. And sickle cell disease patients are 10 times more likely to suffer from seizures than the general population. So seeing something like Zerantin as a very important feature makes sense because it's really commonly prescribed to treat these kinds of seizures. So this gives you a feel for how the 3 different phenotypes that we observed for sickle cell disease when using only clinical data, how they differed, basically. What was it that was driving them apart? Now, let's see what happens if we look at the phenotypes for sickle cell disease that were derived when using the knowledge graph molecular mechanism. So again, 3 subphenotypes, only using mechanism information. What do we see within the first phenotype? Diabetic autonomic neuropathy. So like sickle cell disease, this has been shown to significantly affect the autonomic nervous system. And both disorders have a very significant association with priapism. Aphakia is characterized by the absence of an eye lens and is one of the most commonly caused outcomes of cataract removal surgery. Cataracts are frequently associated with sickle cell disease. So we're getting these explanations from the knowledge graph, and we're also finding literature to justify them. But you can already see it's a different kind of thing that's describing this group of patients. It's more deeply associated with other phenotypes or other diseases which share similarities with these patients. So they can be used to help us describe, well, if there are two different mechanisms that are resulting in the same outcome, then there's an overlap of something important in those mechanisms or those pathways that's really relevant to this subphenotype. Let's look at the next one. So for the second subphenotype, again, using the knowledge graph mechanisms, we find that dabigatran is a really important concept. And what is it? Well, it's an anticoagulant prescribed to sickle cell disease patients that experience venous thromboembolisms. And sickle cell disease patients often experience swollen lymph nodes as a result of chronic infection and histocytosis. So again, instead of being specific medications or really specific types of phenotypes directly associated with sickle cell, like we get from the clinical data, we're getting these deeper associations, more of an underlying genetic component where you're seeing overlap between other things that might explain what you're observing, but not necessarily be directly responsible for sickle cell disease. And the final subphenotype, patients with sickle cell disease are at an increased risk of folate deficiency due to increased erythropoiesis, which can result from neural tube defects like cervical spina bifida. There's a direct association between temporomandibular joint crepitus and sickle cell disease. And so at a high level, the knowledge graph-derived patient representations, again, are going beyond the identification of these surface-level known attributes to define patients, which we get from the clinical data. And it's interesting that we found the same number of subphenotypes, and there definitely are patients that overlapped between them, but there's also different patients that we were able to find from each grouping. And so what do we think about this? That maybe these knowledge graph-derived mechanistic-based representations give us more insight to the actual mechanisms that identify things like relevant drug ingredients, but also relevant similar diseases, things that share common pathways, underlying genetic associations. So they're giving us just this bit more context that we, without molecular information on these patients, could not get just from the clinical data alone. Or at least they're offering a way to explain at a deeper level what we're observing. So diving into the use case, just looking at sickle cell disease. So why did we choose that? So for rare diseases like sickle cell, prevention and treatment differs based on a patient's genotype. Observational data alone have been shown in the literature to be often insufficient to determine the genotype of the patient if there's no newborn screen or molecular data to give that information. So without knowing what someone's genotype is, if all you have is clinical data, it's very difficult to correctly identify the two major genotypes of sickle cell disease. So as a case study, using our best performing molecular mechanism-based patient representations, we wanted to examine whether it was possible to determine the genotypes of our pediatric sickle cell disease patients from our EHR by bringing in data of genotype sickle cell patients, a completely independent population, but similar age, similar disease manifestation, to the same space using the knowledge graph. So if we bring them into the same shared latent space, can we confidently determine which of our clinical patients have which genotype using our genotyped patients? So our first goal was to examine the information available in our knowledge graph for relevant genes to determine how clinically useful patient representations derived from our knowledge graph embeddings were going to be when using only genes, because that's really the only information we had besides the genotype from our geo patients. So the figure on the left is a t-SNE representation of different expressed gene embeddings, which were derived from our phenylator knowledge graph for the geo patients, with the most severe sickle cell disease genotype being HBSS. So we just looked at the most severe genotype because this should be the easiest for us to get useful information. K-means clustering over these embeddings revealed 9 clusters, and the primary single most significant annotation for each of the upregulated differentially expressed genes are labeled on each of the clusters. The second figure shows a t-SNE of the geo differentially expressed gene arrays for the sickle cell genotype. So they're colored by their genotype. Pink and blue, and yellow are the control patients. And each patient's molecular signature was decomposed into a 2-dimensional space. The distance between the average control patient and each of the sickle cell disease patients was found to be significantly associated with a clinical measure of sickle cell severity, such that the molecular signatures of the most severe sickle cell disease patients were the least similar to that of controls. Why do we like that? Well, this gives us good preliminary support that representing genes as embedding arrays to represent these patients actually does capture clinical information such that the most severe sickle cell patients are the least similar to the control patients. So essentially, they're confirming for us that representing sickle cell patients using their genes with some knowledge graph-based information is highly likely to be clinically meaningful. So the Tiffany plots on this slide represent a dimensionality reduced view of the patient representations colored by rare disease type. So you're used to seeing the same colors from our patients before, but we've now added the geo patients using red and gray triangles, where red are the HBSS, the more severe sickle cell genotype, and the gray are the HBSC, the less severe. Patient similarity analyses helped us to confirm that the geo patients were significantly more similar to the EHR-based sickle cell disease patients than they were to any of our other EHR-based rare disease patients. So it's good news in this latent space. The geo patients are closer to our EHR sickle cell patients than they are to any of the rare diseases. The second plot is colored by the clustering assignment, which again we derived using k-means. And the clustering assignment was able to cleanly split the geo sickle cell disease patients into two groups by genotype. So this is a very simple way to try to say, okay, which of our EHR sickle cell disease patients have which genotype, using their similarity or closeness to the different geo-based patient genotypes. And each of them ended up including a similar proportion of patients. So using the clustering assignment to infer the EHR-based patients with sickle cell disease genotypes helped us to actually identify and resolve and correct the genotype for 43 patients from our EHR who we had discovered had diagnoses that were indicative of both genotypes. So they didn't have one clear set of diagnosis codes that pointed to one genotype or the other. So overall, this was a useful task with helping us to say, okay, the knowledge graph is not only really good at helping us to make good, useful patient representations that are distinctive, even when looking at similar diseases, but it also serves as a useful way to take two independent data sets and put them in the same space so that if we're missing information from one, while it might not be perfect and we definitely want some sort of measure of confidence associated, it takes us one step further into giving us more information or insight than we would have had with just the one data source alone. So in conclusion, our second study aimed to understand if a knowledge graph that represented molecular mechanisms of disease could be used to derive meaningful patient representation. And from this, we can conclude the following. So knowledge-based molecular mechanisms resulted in more accurate patient representations using fewer codes than using clinical data alone. The knowledge graph helped us to align independent patient samples with limited to no data in common. And the molecular mechanism-based representations seem to provide a promising first step towards getting towards causality, to having the ability to say, these are the codes that define this patient group, and here's why we think it's right, based off of the underlying biology. And so the primary takeaway from this study was that knowledge graphs are a pretty useful proxy for molecular mechanisms, especially when there's data or information missing from your primary source. And so our grand summary, because both of these studies were definitely not without limitations, what did we learn? It's possible to generate both accurate and performant computational phenotypes using a more automated approach. But it matters what data types you use and what sampling window, and ultimately those things are dictated on the use case of the disease of interest. Using knowledge of the molecular mechanisms that we know underlies human disease can produce potentially clinically actionable computational phenotypes. And because they're implicitly causal, knowledge graphs might help us to explain the logic underlying computational phenotypes, meaning how and why did these patients get put together? Does it make sense clinically? What's the biological rationale? And again, as I mentioned, there are several limitations that are really important to note when interpreting the value of this work. So We had small samples of rare disease patients from a single institution. It would be ideal to look at more rare disease groups, to have bigger sample sizes, and to do this across multiple institutions. We were not able to perform specific chart review beyond confirming with a newborn screen what a primary diagnosis was. So that really does limit the accuracy of how well we can interpret, did we correctly genotype these patients? Kind of. We only used structured EHR data, so some of this missing context we probably could get from the unstructured notes, but they were not available to us at the time of the study. And then our analysis was limited again to a single representation learning method. So there have since been many new methods developed, both for knowledge graphs and for things like text, which is what we primarily use for patient representations, which would be worth exploring to see how they differ, if it changes the outcome of our results, or if we get even better findings. It's really important to note that none of this work would be possible without the guidance, advice, and help of all of the people from all of the different groups shown on this slide. So this was a big undertaking involving many different data sets and different types of domain expertise. And I would also like to thank Paul for the opportunity to present to you today and to share this work, and for your time today as well. And if you have any questions, I'd be happy to take them. And this slide provides some QR codes, the associated GitHub repositories, and then some relevant publications from the 2 resources that I described to you today. And thank you so much for your time.
Thank you so much. That was a great presentation. We have a couple questions from the audience. I have been thinking about some questions as well. Let's start with an audience question from Dr. Stuart Ray. He's wondering about trying to identify, so his example is cystic fibrosis and understanding hyper-specialized care patterns that might be distinct due to practitioner patterns compared to less specialized care.
I'm really glad you brought this up, because there's clearly there's a few holes in the clinical domains that we use, but procedures or visit level information is really important. And we didn't include it. The only reason we didn't initially with this study was there wasn't a good way that we found to incorporate it with the ontology when we were kind of trying to do a one-to-one transfer. We've thought a lot about it since then and talked to extensively people like Melissa Hendel about this, because she also pushes for the inclusion of this sort of procedural information. I don't have a good answer for how we would do that just yet, but I will say I totally acknowledge that it's really important, and I think it would be really valuable for further dividing the subphenotypes, or even helping us to better explain why we're seeing those patients in those groups. There's sort of a different perspective from the knowledge graph, but giving us evidence to justify the groupings.
It is, and I think one of the things that gets me really excited is, so one of my biggest frustrations with the OMOP data model and EHR in general is we know what happened, we don't know why it happened, and we don't often know what the plan was, and so we don't, the inference. And so this idea that, so a patient has a mammogram, they see something suspicious, they end up having a biopsy, the biopsy goes to a pathologist, and then they are diagnosed with cancer, and then you see an ICD-10 code. And so really trying to figure out what was the diagnostic value of the mammogram. So right now in the EHR, they're fairly, and certainly in the OMOP data model, they're unrelated events. They're just temporally sequenced. And so being able to then try to using graph models to determine inference and around why things are happening, I think is very exciting. So do you see applying these techniques for looking for things like diagnostic decision making? Because we focus on treatment value, but I see that there's potential if we can get that inference around diagnostic value.
I would love that, and especially to do it in sort of a time series way, like trajectories to treatment and how those change over time. I think that, certainly using a graph, and now that we've had all this recent developments and improved more efficient computational methods to analyze them, I think that would be a really cool way to approach the problem. I love that idea.
Like your TSNE k-means clustering technique to look for patterns too makes a lot of sense as well. So you give me a little bit of hope that even with the data that we have, which is really transactional in nature, can help us try to infer reason behind it. That is pretty exciting. So we have another question around how do you use a lot of it was kind of clustering. Are you using any distance measures between your concepts as part of your network analysis?
Certainly, in not directly in terms of like how the embeddings are derived for the graph, although there is some component of that in these methods. I primarily use them on the classification side. So being able to calculate cosine similarity between the patient vectors and to sort of use that with the Uden index to determine, is this patient more like, does this patient look like this other patient? Did we correctly classify them to group them? When you're in this latent space, the distant metrics are not only really informative for a pretty cheap computation, but they are easy to translate quickly between different data types as well. So they make sense when you talk to someone, especially a doctor or a biologist, when you can tell them what that means, and it's easier to talk about that than some of the other things we use in modeling. So we definitely use them, not necessarily in the network analysis yet. They clearly have value there. If we stayed in the traditional graph traversing based approaches, then yes, shortest paths and other types of search, which are based off of distance, will be very valuable. We hopped out of that because the graph was so big. So we primarily use them for patient classification.
Harold, did you have a question or comment?
Yeah, so Tiffany, this is quite amazing. I'm kind of dizzy from all the many levels that you're going through, and I'm a pretty abstract guy, so that's very impressive. I have two questions. One is kind of concrete, one less. The concrete question, in sickle cell disease, which I've been somewhat involved in, we make a big deal about neurological symptoms and strokes and silent strokes, and yet I didn't see, I mean, the anticoagulant is a hint, but there's nothing about transfusion, there's nothing about MRIs, nothing about Dopplers. I was a little surprised or maybe you just didn't have that data in your data set.
The way that the experts I was working with wanted me to show the most important features were not just the actual most important features, but the most important features that were unique to each of the clusters specifically. So we definitely did have the specific things that you're mentioning, but if they weren't unique, as in the things that were distinct between the subphenotypes of a given disease, we sort of set them aside for that initial analysis.
Clinically, we say, oh, these kids are the severe kids who have repeated episodes and stuff like that. So again, maybe when you put time in. The more abstract question is, so the reason you do research is about things that you're uncertain about, right? And knowledge graphs have this false sense of knowledge, which some of us probabilists are a little. So where's the uncertainty in all this?
In terms of,
well, you're doing the research in order to represent an uncertainty, right?
So
and
where?
So how where does this saying here are some associations, but we're not really sure. So where does that uncertainty come in?
So yes, and from the beginning, from the way we represent patients. So I think, let me answer this in one way, and then I'll try to answer it in a different way. So from the clinical side, we use codes that are distinctive to find patients, to identify who are our patients in each disease, but we then exclude everything that is known and distinct and used in formal definitions for phenotypes to find sickle cell patients when we actually build the representation. So their representation shows everything else about a sickle cell patient that isn't the primary diagnosis of sickle cell disease or the well-known frequently occurring things that you would use to find them. So the lab tests and the different procedures. So there's some uncertainty in looking at the question of, well, how well can we do at identifying and correctly classifying these patients when we don't have the things that should make them really easy to classify? So what are the other clinical aspects about them? And then we also get that in the knowledge graph from the perspective that not all of the concepts, so there's fewer concepts used when you're taking the knowledge graph embeddings and aligning them to the clinical codes, because that information just isn't in the graph. So there might not actually be a specific code that's really useful for sickle cell disease in our knowledge graph. So there's also some missing information or uncertainty around, can we actually represent and find these patients when we are missing known significant phenotypes or diseases of these patients? And I think the biggest form of uncertainty really lies around that last use case is how well can we bring in independent patient populations? So they're similar in age, they're similar in gender. We don't know anything else about them. Let's say in this case from our geo patients clinically, how good does the knowledge graph mechanism representation, how good is it at actually helping us correctly group patients together on an attribute that we don't have in the EHR? So I think we have uncertainty from the fact that we purposefully held out information we know would make us more certain in our groupings. And we want to determine, well, is it still useful and what is it that's driving that? Same on the mechanism side, we're definitely missing things that are important. We excluded the other things on purpose. What else on the graph, if it's connected to, is useful and how useful is it? And then what's the value of this latent space of shared information to bringing in independent data sets, independent patients for creating a more informative view, if you will, of a patient or a disease? So I don't know if I directly answered your question, but I hope I hinted at some forms of uncertainty.
And we do have another question, but I'll leave the Bayesian question for later.
Harold, did you want to ask Star's question?
Oh, I was actually paying attention to Tiffany, so I couldn't read it. Go ahead, Paul. I can't.
So yeah, the other question we have for you was, depending on the use case, non-clinical and non-genetic causes may be of value. How could things like social determinants of health enhance reasoning? Perhaps things would differ not only based on diseases, but also based on different decisions further downstream. Then the question is how accurate or valuable the graph models are for decision-making, or how much alignment there is with decision-making. I'm not quite sure it makes sense.
Social determinants of health, absolutely important. And it was unfortunate that towards the end of my PhD at the hospital I was working with to have access to the data was willing to share more towards the end, and we just didn't get to including them. But we did get to a point where we could group patients by the last or first few digits of their zip code. So a general broad view, and in certain areas, like we know from Denver where we were, care definitely differs. Just the availability of resources for care differs significantly. If you're out in the countryside, it's really hard to get specialized treatment. You're showing up not very often at Children's, which is a specialized center where we took our data from, versus places in Denver proper where urgent care might just be 2 blocks down the road. And there are more than that, but we definitely know it matters. And it's definitely something that I so badly wish we could have included, especially more demographics are difficult and fragile with EHR data. So I did use them in one version. I didn't include it in this analysis because we just didn't feel confident about the bias that we were introducing into our analysis. So totally hear you, 100% agree that that's really valuable. And I do think it would be really informative for helping to either create different groupings or definitely offer another layer of explanation into what we're observing. In terms of using graphs for decision-making, I think your question, and please correct me if I'm wrong, was how useful do I think they will be, especially if they're built, in our case, not from clinical data, but they share similarities with clinical data. I think should you get practitioners who are open to using them and trust the resource and a system that could use it, I think it could be, I mean, the immediate most obvious answer in my mind is it could be something that could assist things like UpToDate. So these resources which clinicians use to help make decisions would provide additional information. I think you could use it as another source of information. Assuming you had confidence, you had some kind of score to help the person using it to know how much they should trust it. I think it could be really valuable for making really deep or interesting connections between things that may otherwise not be easy to connect. So the ability to have 700,000 nodes and 25 million edges connected between them gives you a different view of the world than you have when you're looking at those data sources separately. So as to exactly what the benefit of that is or how it could impact diagnostic or clinical care, I'm not going to speak to that, but I hope and think it could be really valuable if it was built in a trustworthy way and was reliable.
The way I would see the question is that the further downstream you go, down into the disease, obviously you can gain a lot of value for understanding the disease. However, when you try to then use that upstream as a diagnostic tool to actually implement or affect decision support, then you have to go to probabilistic models or RAP to be able to implement them. And so I actually feel there's a ton of value for going upstream and downstream. So we certainly love. And I like this idea of trying to find diagnostic errors or diagnostic decision making that we can then we call it a retrospective scope, where we know what happened to the patient, and so we can kind of then see what sort of decisions have been made at a certain point in time. And so I think this I think there's enormous value for trying to understand it, but then where we actually deploy the decision support tools in care becomes critical for what information we know at that time to be able to inform potential decision making from clinicians.
Absolutely. You said it way better than I did.
So Tiffany, thank you so much for your time. That was such a great conversation. It's really exciting to see where this is developing. I really enjoy. I really am glad we're starting to begin to integrate mechanistic models into transactional data. And thank you again for a wonderful conversation.
Thank you so much, and thanks to everyone for your time and just for the opportunity to be here. I really enjoyed it.
Yeah, thank you. And that concludes our grand rounds. Have a great day, everyone. Thank you.